Sympatric speciation. In evolutionary biology, sympatric speciation is the evolution of a new species from a surviving ancestral species while both continued to inhabit the same geographic region. In evolutionary biology and biogeography, sympatric and sympatry are terms referring to organisms whose ranges overlap so that they occur together at least in some places. If these organisms are closely related, e.g. sister species, such a distribution may be the result of sympatric speciation. Etymologically, sympatry is derived from Greek syn, son, together, and patris, fatherland. 1. The term was coined by Edward Bagnall Pelton in 1904, who explains the derivation. 1. In sympatric speciation, reproductive isolation evolves within a population without the aid of geographic barriers. Sympatric speciation is one of three traditional geographic modes of speciation. 2. 3. Allopatric speciation is the evolution of species caused by the geographic isolation of two or more populations of a species. In this case, divergence is facilitated by the absence of gene flow. Parapatric speciation is the evolution of geographically adjacent populations into distinct species. In this case, divergence occurs despite limited interbreeding where the two diverging groups come into contact. In sympatric speciation, there is no geographic constraint to interbreeding. These categories are special cases of a continuum from zero sympatric to complete allopatric spatial segregation of diverging groups. 3. In multicellular eukaryotic organisms, sympatric speciation is a plausible process that is known to occur, but the frequency with which it occurs is not known. 4. In bacteria, however, the analogous process defined as the origin of new bacterial species that occupy definable ecological niches might be more common because bacteria are less constrained by the homogenizing effects of sexual reproduction and are prone to comparatively dramatic and rapid genetic change through horizontal gene transfer. 5. Evidence. Sympatric speciation events are quite common in plants, which are prone to acquiring multiple homologous sets of chromosomes, resulting in polyploidy. The polyploid offspring occupy the same environment as the parent plants, hence symmetry, but are reproductively isolated. A number of models have been proposed for alternative modes of sympatric speciation. The most popular, which invokes the disruptive selection model, was first put forward by John Maynard Smith in 1966. 6. Maynard Smith suggested that homozygous individuals may, under particular environmental conditions, have a greater fitness than those with alleles heterozygous for a certain trait. Under the mechanism of natural selection, therefore, homozygosity would be favored over heterozygosity, eventually leading to speciation. Sympatric divergence could also result from a sexual conflict. 7. Disruption may also occur in multiple gene traits. The medium brown finch Geospiza fortis is showing gene pool divergence in a population on Santa Cruz Island. Beak morphology conforms to two different size ideals, while intermediate individuals are selected against. Some characteristics termed magic traits, such as beak morphology, may drive speciation because they also affect mating signals. In this case, different beak phenotypes may result in different bird calls, providing a barrier to exchange between the gene pools. 8. A somewhat analogous system has been reported in horseshoe bats, in which echolocation call frequency appears to be a magic trait. In these bats, the constant frequency component of the call not only determines prey size but may also function in aspects of social communication. Work from one species, the large-eared horseshoe bat Rhinolophus philippinensis, shows that abrupt changes in call frequency among sympatric morphs is correlated with reproductive isolation. 9. A further well-studied circumstance of sympatric speciation is when insects feed on more than one species of host plant. In this case, insects become specialized as they struggle to overcome the various plants' defense mechanisms. Dress and Mallet, 2002-10. Rhagoletis pomonella, the apple maggot, may be currently undergoing sympatric or, more precisely, heteropatric speciation. The apple feeding race of this species appears to have spontaneously emerged from the hawthorn feeding race in the 1800-1850 AD time frame, after apples were first introduced into North America. The apple feeding race does not now normally feed on hawthorns, and the hawthorn feeding race does not now normally feed on apples. This may be an early step towards the emergence of a new species. 11, 12, 13. Some parasitic ants may have evolved via sympatric speciation. 14. Isolated and relatively homogeneous habitats such as crater lakes and islands are among the best geographical settings in which to demonstrate sympatric speciation. For example, Nicaragua crater lake cichlid fishes include 9 described species and dozens of undescribed species that have evolved by sympatric speciation. 15. 16. Monostromal latissimum, a marine green algae, also shows sympatric speciation in southwest Japanese islands. Although panmictic, the molecular phylogenetics using nuclear introns revealed staggering diversification of population. 17. African cichlids also offer some evidence for sympatric speciation. They show a large amount of diversity in the African Great Lakes. Many studies point to sexual selection as a way of maintaining reproductive isolation. Female choice with regards to male coloration is one of the more studied modes of sexual selection in African cichlids. Female choice is present in cichlids because the female does much of the work in raising the offspring, while the male has little energy input in the offspring. She exerts sensory bias when picking males by choosing those that have colors similar to her or those that are the most colorful. 18, 19, 20. This helps maintain sympatric speciation within the lakes. Cichlids also use acoustic reproductive communication. The male cichlid quivers as a ritualistic display for the female which produces a certain number of pulses and pulse period. Female choice for good genes and sensory bias is one of the deciding factors in this case, selecting for calls that are within her species and that give the best fitness advantage to increase the survivability of the offspring. 21, 22. Male-male competition is a form of intersexual selection and also has an effect on speciation in African cichlids. Ritualistic fighting among males establishes which males are going to be more successful in mating. This is important in sympatric speciation because species with similar males may be competing for the same females. There may be a fitness advantage for one phenotype that could allow one species to invade another. 23, 24. Studies show this effect in species that are genetically similar, have the capability to interbreed, and show phenotypic color variation. Ecological character displacement is another means for sympatric speciation. Within each lake there are different niches that a species could occupy. For example, different diets and depth of the water could help to maintain isolation between species in the same lake. Allochrony offers some empirical evidence that sympatric speciation has taken place, as many examples exist of recently diverged, sister taxa, allochronic species. A case of ongoing sympatric divergence due to allochrony might be found in the marine insect Clunio marinus. 25. A rare example of sympatric speciation in animals is the divergence of resident and transient orca forms in the Northeast Pacific. 26. Resident and transient orcas inhabit the same waters, but avoid each other and do not interbreed. The two forms hunt different prey species and have different diets, vocal behavior, and social structures. Some divergences between species could also result from contrasts in microhabitats. A population bottleneck occurred around 200,000 years ago, greatly reducing the population size at the time as well as the variance of genes, which allowed several ecotypes to emerge afterwards. 27. The European polecat Mustela puturius exhibited a rare dark phenotype similar to the European mink Mustela lutriola phenotype, which is directly influenced by peculiarities of forest brooks. 28. Controversy. For some time it was difficult to prove that sympatric speciation was possible, because it was impossible to observe it happening. 3. It was believed by many, and championed by Ernst Mayr, that the theory of evolution by natural selection could not explain how two species could emerge from one if the subspecies were able to interbreed. 29. Since Mayer's heyday in the 1940s and 50s, mechanisms have been proposed that explain how speciation might occur in the face of interbreeding, also known as gene flow. 30. And even more recently, concrete examples of sympatric divergence have been empirically studied. 31. 32. The debate now turns to how often sympatric speciation may actually occur in nature and how much of life's diversity it may be responsible for. History. The German evolutionary biologist Ernst Mayr argued in the 1940s that speciation cannot occur without geographic, and thus reproductive, isolation. 29. He stated that gene flow is the inevitable result of symmetry, which is known to squelch genetic differentiation between populations. Thus, a physical barrier must be present, he believed, at least temporarily, in order for a new biological species to arise. 33. This hypothesis is the source of much controversy around the possibility of sympatric speciation. Mayer's hypothesis was popular and consequently quite influential, but is now widely disputed. 34. The first to propose what is now the most pervasive hypothesis on how sympatric speciation may occur was John Maynard Smith, in 1966. He came up with the idea of disruptive selection. He figured that if two ecological niches are occupied by a single species, diverging selection between the two niches could eventually cause reproductive isolation. 35. By adapting to have the highest possible fitness in the distinct niches, two species may emerge from one even if they remain in the same area, and even if they are mating randomly. 30. Defining symmetry. Investigating the possibility of sympatric speciation requires a definition thereof, especially in the 21st century, when mathematical modeling is used to investigate or to predict evolutionary phenomena. 34. Much of the controversy concerning sympatric speciation may lie solely on an argument over what sympatric divergence actually is. The use of different definitions by researchers is a great impediment to empirical progress on the matter. The dichotomy between sympatric and allopatric speciation is no longer accepted by the scientific community. It is more useful to think of a continuum, on which there are limitless levels of geographic and reproductive overlap between species. On one extreme is allopatry, in which the overlap is zero, no gene flow, and on the other extreme is sympatry, in which the ranges overlap completely, maximal gene flow. The varying definitions of sympatric speciation fall generally into two categories: definitions based on biogeography, or on population genetics. As a strictly geographical concept, sympatric speciation is defined as one species diverging into two while the ranges of both nascent species overlap entirely. This definition is not specific enough about the original population to be useful in modeling. 3. Definitions based on population genetics are not necessarily spatial or geographical in nature, and can sometimes be more restrictive. These definitions deal with the demographics of a population, including allele frequencies, selection, population size, the probability of gene flow based on sex ratio, life cycles, etc. The main discrepancy between the two types of definitions tends to be the necessity for panmixia. Population genetics definitions of symmetry require that mating be dispersed randomly, or that it be equally likely for an individual to mate with either subspecies, in one area as another, or on a new host as a nascent one. This is also known as panmixia. 3. Population genetics definitions, also known as non-spatial definitions, thus require the real possibility for random mating, and do not always agree with spatial definitions on what is and what is not symmetry. For example, microallopatry, also known as macrosymmetry, is a condition where there are two populations whose ranges overlap completely, but contact between the species is prevented because they occupy completely different ecological niches, such as diurnal versus nocturnal. This can often be caused by host-specific parasitism, which causes dispersal to look like a mosaic across the landscape. Microallopatry is included as symmetry according to spatial definitions, but, as it does not satisfy panmixia, it is not considered symmetry according to population genetics definitions. 3. Mallet et al. 2002 claims that the new non-spatial definition is lacking in an ability to settle the debate about whether sympatric speciation regularly occurs in nature. They suggest using a spatial definition, but one that includes the role of dispersal, also known as cruising range, so as to represent more accurately the possibility for gene flow. They assert that this definition should be useful in modeling. They also state that under this definition, sympatric speciation seems plausible. 33. Current state of the controversy. Evolutionary theory as well as mathematical models have predicted some plausible mechanisms for the divergence of species without a physical barrier. 30. In addition, there have now been several studies that have identified speciation that has occurred, or is occurring with gene flow, see section above, evidence. Molecular studies have been able to show that, in some cases where there is no chance for allopatry, species continue to diverge. One such example is a pair of species of isolated desert palms. Two distinct, but closely related species exist on the same island, but they occupy two distinct soil types found on the island, each with a drastically different pH balance. 31. Because they are palms, they send pollen through the air, they could freely interbreed, except that speciation has already occurred, so that they do not produce viable hybrids. This is hard evidence for the fact that, in at least some cases, fully sympatric species really do experience diverging selection due to competition, in this case for a spot in the soil. This, and the other few concrete examples that have been found, are just that, they're few, so they tell us little about how often sympatry actually results in speciation in a more typical context. The burden now lies on providing evidence for sympatric divergence occurring in non-isolated habitats. It is not known how much of the Earth's diversity it could be responsible for. Some still say that panmixia should slow divergence, and thus sympatric speciation should be possible but rare. 1. Meanwhile, others claim that much of the Earth's diversity could be due to speciation without geographic isolation. 36. The difficulty in supporting a sympatric speciation hypothesis has always been that an allopatric scenario could always be invented, and those can be hard to rule out, but with modern molecular genetic techniques can be used to support the theory. 36. In 2015, cichlid fish from a tiny volcanic crater lake in Africa were observed in the act of sympatric speciation using DNA sequencing methods. A study found a complex combination of ecological separation and mate choice preference had allowed two ecomorphs to genetically separate even in the presence of some genetic exchange. 37. 38. Environment portal. Ecology portal. Earth sciences portal. Evolutionary biology portal. Heteropatric speciation is a special case of sympatric speciation that occurs when different ecotypes or races of the same species geographically coexist but exploit different niches in the same patchy or heterogeneous environment. It is thus as a refinement of sympatric speciation, with a behavioral, rather than geographical barrier to the flow of genes among diverging groups within a population. Behavioral separation is a mechanism for promoting sympatric speciation in a heterogeneous, or patchwork landscape, was highlighted in John Maynard Smith's seminal paper on sympatric speciation. 39. In recognition of the importance of this behavioral versus geographic distinction, Wayne Getz and Vallejo Kaidala introduced the term heteropatry in their extension of Maynard Smith's analysis 40 of conditions that facilitate sympatric speciation. Although some evolutionary biologists still regard sympatric speciation as highly contentious, both theoretical 41 and empirical 42 studies support it as a likely explanation of the diversity of life in particular ecosystems. Arguments implicate competition and niche separation of sympatric ecological variants that evolve through assortative mating into separate races and then species. Assortative mating most easily occurs if mating is linked to niche preference, as occurs in the apple maggot Rhagoletis pominella, where individual flies from different races use volatile odors to discriminate between hawthorn and apple and look for mates on natal fruit. The term heteropatry semantically resolves the issue of sympatric speciation by reducing it to a scaling issue in terms of the way the landscape is used by individuals versus populations. From a population perspective, the process looks sympatric, but from an individual's perspective, the process looks allopatric, once the time spent flying over or moving quickly through intervening non-preferred niches is taken into account. Adaptive radiation cladistics. Ecotype. History of speciation, hybrid speciation, phylogenetics. Polymorphism, biology. Heteropatric speciation. See also. Tiger. Project Tiger is a wildlife conservation movement initiated in India to protect the endangered tiger. The project was initiated in 1973 by the Ministry of Environment, Forest and Climate Change of the Government of India. As of March 2024, there are 55 protected areas that have been designated as tiger reserves under the project. As of 2023, there were 3,682 wild tigers in India, which is almost 75% of the world's wild tiger population. Project Tiger was initiated in 1973 by the Ministry of Environment, Forest and Climate Change of the Government of India. The project was initiated to protect the Bengal tiger and its habitats and to establish dedicated tiger reserves for sustaining tiger populations. 1. As per the Section 38 of the Wildlife Protection Act, 1972, the state governments are responsible for preparing a tiger conservation plan which includes planning and management of notified areas and maintaining the requisite competent staff to ensure the protection of the tiger reserve and providing inputs for maintaining a viable population of tigers, co-predators and prey animals. 2. 3. Tiger reserves consist of a core area which includes parts of protected areas such as a national park or a wildlife sanctuary and a buffer zone which is a mix of forested and non-forested land. Project Tiger is aimed at performing the necessary activities to ensure viability of tiger population in the core area and to promote a balance between the existence of people and animals in the buffer zones. 1. In 2006, National Tiger Conservation Authority was formed to administer the tiger reserves which were set up as a part of Project Tiger, with Project Tiger becoming a centrally sponsored scheme, CSS, to provide funding for the establishment and administration of the tiger reserves. 1. In 1973, 9 protected areas were initially designated as tiger reserves. By the late 1980s, the initial 9 reserves covering an area of 9,115 square kilometers, 3,519 square miles, had been increased to 15 reserves. Project Tiger. Banner logo of Project Tiger. Tiger Conservation Program Overview. Formed the 1st of April 1973. Motto: India leads tiger conservation. Parent Department: National Tiger Conservation Authority. Website. History and objectives. Covering an area of 24,700 square kilometers, 9,500 square miles. 4. By 1997, 23 tiger reserves encompassed an area of 33,000 square kilometers, 13,000 square miles. 5. As of March 2024, there are 55 protected areas that have been designated as tiger reserves. 2. Tiger population. State-wise tiger population 2018. During the tiger census of 2006, a new methodology was used extrapolating site-specific densities of tigers, their co-predators and prey derived from camera trap and sign surveys using GIS. Based on the result of these surveys, the total tiger population was estimated at 1,411 individuals ranging from 1,165 to 1,657 adult and sub-adult tigers of more than 1.5 years of age. 6th 7th The 2010 National Tiger Assessment estimated the total population of wild tigers in India at 1,706. As per Ministry of Environment and Forests, the wild tiger population in India stood at 2,226 in 2014 with an increase of 30.5% since the 2010 estimate. 8. In 2018, according to the National Tiger Conservation Authority, there were an estimated 3,346 wild tigers with an average of 2,967 in existence in India. 9th The wild tiger population increased to 3,682 as of 2022. 10. As India is home to majority of the global wild tiger population, the increase in population of tigers in India played a major role in driving up global populations as well. The number of wild tigers globally rose from 3,159 in 2010 to 3,890 in 2016 according to the Worldwide Fund and Global Tiger Forum. 11. Management and administration. A Bengal tiger in Mudumalai Tiger Reserve. Project Tiger is headed by an Additional Director General (ADG) based at New Delhi with regional offices at Bangalore, Guwahati and Nagpur. 1. The wildlife habitats that fall under Project Tiger are categorized into different conservation units: Shivalik Terai, Northeast, Sundarbans, Western Ghats, Eastern Ghats, Central India and Sariska. 12. Function under the ambit of Project Tiger include protection of tiger habitats, daily monitoring, facilitating ecological development for local people in the buffer zones, voluntary relocation of people from core critical tiger habitats and addressing human-wildlife conflicts. As a part of the project, Stater provided assistance on curtailing poaching activities such as disseminating information on poachers, assisting in combing forest floor to check for traps and other anti-poaching activities, maintaining tiger database, providing grants and training for deployment of Special Tiger Protection Force. 13. Wireless communication systems, infrared thermal cameras and monitoring systems have been developed within the tiger reserves to assist in patrol activities. 14. 15. Challenges. Project Tiger's efforts are hampered by poaching, deforestation, construction and irregularities in administration of certain reserves. 16, 17, 18 The Forest Rights Act enacted by the Indian government in 2006 recognizes the rights of forest-dwelling communities in the buffer zones. Some of the wildlife experts have questioned the implications of the same on tiger conservation as it will increase human-animal conflict and might give opportunities for poaching. 19, 20 While others argue that this overlooks the reality of human-tiger coexistence and the abuse of power by authorities wherein local people who have been coexisting with the animals being evicted from their traditional lands rather than allowing them a proper role in decision-making to aid the tiger crisis. 21, 22 Construction and irregularities in administration of certain reserves. 16, 17, 18 The Forest Rights Act enacted by the Indian government in 2006 recognizes the rights of forest-dwelling communities in the buffer zones. Some of the wildlife experts have questioned the implications of the same on tiger conservation as it will increase human-animal conflict and might give opportunities for poaching. 19, 20 While others argue that this overlooks the reality of human-tiger coexistence and the abuse of power by authorities wherein local people who have been coexisting with the animals being evicted from their traditional lands rather than allowing them a proper role in decision-making to aid the tiger crisis. 21. An ecosystem model is an abstract, usually mathematical, representation of an ecological system ranging in scale from an individual population to an ecological community, or even an entire biome which is studied to better understand the real system. 2. A structural diagram of the open ocean plankton ecosystem model of Fasham, Duclos and McKelvey, 1990. 1. Using data gathered from the field, ecological relationships, such as the relation of sunlight and water availability to photosynthetic rate, or that between predator and prey populations, are derived, and these are combined to form ecosystem models. These model systems are then studied in order to make predictions about the dynamics of the real system. Often, the study of inaccuracies in the model, when compared to empirical observations, will lead to the generation of hypotheses about possible ecological relations that are not yet known or well understood. Models enable researchers to simulate large-scale experiments that would be too costly or unethical to perform on a real ecosystem. They also enable the simulation of ecological processes over very long periods of time, i.e. simulating a process that takes centuries in reality, can be done in a matter of minutes in a computer model. 3. Ecosystem models have applications in a wide variety of disciplines, such as natural resource management, 4. Ecotoxicology and environmental health, 5. 6. Agriculture, 7. And wildlife conservation. 8. Ecological modeling has even been applied to archaeology with varying degrees of success, for example, combining with archaeological models to explain the diversity and mobility of stone tools. 9. Types of models. There are two major types of ecological models, which are generally applied to different types of problems: 1. Analytic models and 2. Simulation computational models. Analytic models are typically relatively simple, often linear, systems that can be accurately described by a set of mathematical equations whose behavior is well known. Simulation models, on the other hand, use numerical techniques to solve problems for which analytic solutions are impractical or impossible. Simulation models tend to be more widely used, and are generally considered more ecologically realistic, while analytic models are valued for their mathematical elegance and explanatory power. 10, 11, 12 EcoPath is a powerful software system which uses simulation and computational methods to model marine ecosystems. It is widely used by marine and fisheries scientists as a tool for modeling and visualizing the complex relationships that exist in real-world marine ecosystems. 13, 14, 15, 16, 17, 18, 19. Model design. Diagram of the Silver Springs Model, Odum, 1971. Note the aggregation into functional groups such as herbivores or decomposers. 20. The process of model design begins with a specification of the problem to be solved and the objectives for the model. 21. Ecological systems are composed of an enormous number of biotic and abiotic factors that interact with each other in ways that are often unpredictable, or so complex as to be impossible to incorporate into a computable model. Because of this complexity, ecosystem models typically simplify the systems they are studying to a limited number of components that are well understood and deemed relevant to the problem that the model is intended to solve. 22, 23. The process of simplification typically reduces an ecosystem to a small number of state variables and mathematical functions that describe the nature of the relationships between them. 24. The number of ecosystem components that are incorporated into the model is limited by aggregating similar processes and entities into functional groups that are treated as a unit. 25, 26. After establishing the components to be modeled and the relationships between them, another important factor in ecosystem model structure is the representation of space used. Historically, models have often ignored the confounding issue of space. However, for many ecological problems, spatial dynamics are an important part of the problem, with different spatial environments leading to very different outcomes. Spatially explicit models, also called spatially distributed or landscape models, attempt to incorporate a heterogeneous spatial environment into the model. 27, 28, 29. A spatial model is one that has one or more state variables that are a function of space, or can be related to other spatial variables. 30. Validation. After construction, models are validated to ensure that the results are acceptably accurate or realistic. One method is to test the model with multiple sets of data that are independent of the actual system being studied. This is important since certain inputs can cause a faulty model to output correct results. Another method of validation is to compare the model's output with data collected from field observations. Researchers frequently specify beforehand how much a disparity they are willing to accept between parameters output by a model and those computed from field data. 31, 32, 33, 34, 35. Examples. The Lotka-Volterra equations. A sample time series of the Lotka-Volterra model. Note that the two populations exhibit cyclic behavior, and that the predator cycle lags behind that of the prey. One of the earliest, 36, and most well-known ecological models is the predator-prey model of Alfred J. Lotka, 1925-37, and Vito Volterra, 1926. 38. This model takes the form of a pair of ordinary differential equations, one representing a prey species, the other its predator. Where is the number concentration of the prey species? Is the number concentration of the predator species? Is the prey species growth rate? Is the predation rate of upon? Is the assimilation efficiency of? Is the mortality rate of the predator species? Volterra originally devised the model to explain fluctuations in fish and shark populations observed in the Adriatic Sea after the First World War, when fishing was curtailed. However, the equations have subsequently been applied more generally. 39. Although simple, they illustrate some of the salient features of ecological models. Modeled biological populations experience growth, interact with other populations as either predators, prey or competitors, and suffer mortality. A credible, simple alternative to the Lotka-Volterra predator-prey model and its common prey-dependent generalizations is the ratio-dependent or Arditi-Ginzburg model. 40. The two are the extremes of the spectrum of predator interference models. According to the authors of the alternative view, the data show that true interactions in nature are so far from the Lotka-Volterra extreme on the interference spectrum that the model can simply be discounted as wrong. They are much closer to the ratio-dependent extreme, so if a simple model is needed, one can use the Arditi-Ginzburg model as the first approximation. 41. Others. The theoretical ecologist Robert Ulanowitz has used information theory tools to describe the structure of ecosystems, emphasizing mutual information correlations in studied systems. Drawing on this methodology and prior observations of complex ecosystems, Ulanowitz depicts approaches to determining the stress levels on ecosystems and predicting system reactions to define types of alteration in their settings, such as increased or reduced energy flow and eutrophication. 42. Conway's Game of Life and its variations model ecosystems where proximity of the members of a sequence analysis. In bioinformatics, sequence analysis is the process of subjecting a DNA, RNA or peptide sequence to any of a wide range of analytical methods to understand its features, function, structure, or evolution. It can be performed on the entire genome, transcriptome or proteome of an organism, and can also involve only selected segments or regions, like tandem repeats and transposable elements. Methodologies used include sequence alignment, searches against biological databases, and others. 1. Since the development of methods of high-throughput production of gene and protein sequences, the rate of addition of new sequences to the databases increased very rapidly. Such a collection of sequences does not, by itself, increase the scientist's understanding of the biology of organisms. However, comparing these new sequences to those with known functions is a key way of understanding the biology of an organism from which a new sequence comes. Thus, sequence analysis can be used to assign function to coding and non-coding regions in a biological sequence, usually by comparing sequences and studying similarities and differences. Nowadays, there are many tools and techniques that provide the sequence comparisons, sequence alignment, and analyze the alignment product to understand its biology. Sequence analysis in molecular biology includes a very wide range of processes. 1. The comparison of sequences to find similarity, often to infer if they are related homologous. 2. Identification of intrinsic features of the sequence such as active sites, post-translational modification sites, gene structures, reading frames, distributions of introns and exons and regulatory elements. 3. Identification of sequence differences and variations such as point mutations and single nucleotide polymorphism, SNP, in order to get the genetic marker. 4. Revealing the evolution and genetic diversity of sequences and organisms. 5. Identification of molecular structure from sequence alone. History. Since the very first sequences of the insulin protein were characterized by Fred Sanger in 1951, biologists have been trying to use this knowledge to understand the function of molecules. 2. He and his colleagues' discoveries contributed to the successful sequencing of the first DNA-based genome. 4. The method used in this study, which is called the Sanger method, or Sanger sequencing, was a milestone in sequencing long-strand molecules such as DNA. This method was eventually used in the Human Genome Project. 5. According to Michael Levitt, sequence analysis was born in the period from 1969 to 1977. 6. In 1969, the analysis of sequences of transfer RNAs was used to infer residue interactions from correlated changes in the nucleotide sequences, giving rise to a model of the tRNA secondary structure. 7. In 1970, Saul B. Needleman and Christian D. Wunsch published the first computer algorithm for aligning two sequences. 8. Over this time, developments in obtaining nucleotide sequence improved greatly, leading to the publication of the first complete genome of a bacteriophage in 1977. 9. Robert Hawley and his team in Cornell University were believed to be the first to sequence an RNA molecule. 10. Overview of nucleotide sequence analysis, DNA and RNA. Nucleotide sequence analyses identify functional elements like protein binding sites, uncover genetic variations like SNPs, study gene expression patterns, and understand the genetic basis of traits. It helps to understand mechanisms that contribute to processes like replication and transcription. Some of the tasks involved are outlined below. Quality control and preprocessing. Quality control assesses the quality of sequencing reads obtained from the sequencing technology, e.g. Illumina. It is the first step in sequence analysis to limit wrong conclusions due to poor quality data. The tools used at this stage depend on the sequencing platform. For instance, FastQC checks the quality of short reads, including RNA sequences. Nanoplot or PycoQC are used for long read sequences, e.g. Nanopore sequence reads, and MultiQC aggregates the result of FastQC in a web page format. 11, 12, 13. Quality control provides information such as read lengths, GC content, presence of adapter sequences for short reads, and a quality score, which is often expressed on a Phred scale. 14. If adapters or other artifacts from PCR amplification are present in the reads, particularly short reads, they are removed using software such as Trimmomatic. 15. Or QtAdapt. 16. Read alignment. At this step, sequencing reads whose quality have been improved are mapped to a reference genome using alignment tools like BWA. 17. For short DNA sequence reads, Minimap. 18. For long read DNA sequences, and START. 19. For RNA sequence reads. The purpose of mapping is to find the origin of any given read based on the reference sequence. It is also important for detecting variations or phylogenetic studies. The output from this step, that is, the aligned reads, are stored in compatible file formats known as SAM, which contains information about the reference genome as well as individual reads. Alternatively, BAM file formats are preferred as they use much less disk or storage space. 14. Note, this is different from sequence alignment, which compares two or more whole sequences, or sequence regions, to quantify similarity or differences or to identify an unknown sequence, as discussed below. The following analysis steps are peculiar to DNA sequences. Variant calling. Identifying variants is a popular aspect of sequence analysis, as variants often contain information of biological significance, such as explaining the mechanism of drug resistance in an infectious disease. These variants could be single nucleotide variants, SNVs, small insertions, deletions, indels, and large structural variants. The read alignments are sorted using SAM rules, after which variant callers such as GATK20 are used to identify differences compared to the reference sequence. The choice of variant calling tool depends heavily on the sequencing technology used, so GATK is often used when working with short reads, while long read sequences require tools like Deep Variant 21 and Sniffles. 22. Tools may also differ based on organism, prokaryotes or eukaryotes, source of sequence data, cancer versus metagenomic, and variant type of interest, SNVs or structural variants. The output of variant calling is typically in VCF format, and can be filtered using allele frequencies, quality scores, or other factors based on the research question at hand. 14. Variant annotation. This step adds context to the variant data using curated information from peer-reviewed papers and publicly available databases like NAM-AD and Ensembl. Variants can be annotated with information about genomic features, functional consequences, regulatory elements, and population frequencies using tools like INAVAR or SNFF, 23, or custom scripts and pipeline. The output from this step is an annotation file in BED or TXT format. 14. Visualization and interpretation. Genomic data, such as read alignments, coverage plots, and variant calls, can be visualized using genome browsers like IGV, Integrative Genomics Viewer, or UCSC Genome Browser. Interpretation of the results is done in the context of the biological question or hypothesis under investigation. The output can be a graphical representation of data in the forms of Sersos plots, volcano plots, etc., or other forms of report describing the observations. 14. DNA sequence analysis could also involve statistical modeling to infer relationships in epigenetic analysis, like identifying differential methylation regions using a tool like DSS. The following steps are peculiar to RNA sequences. Gene expression analysis. Mapped RNA sequences are analyzed to estimate gene expression levels using quantification tools such as HTSEQ24 and identify differentially expressed genes (DEGs) between experimental conditions using statistical methods like DESEQ2. 25. This is carried out to compare the expression levels of genes or isoforms between or across different samples and infer biological relevance. 14. The output of gene expression analysis is typically a table with values representing the expression levels of gene IDs or names in rows and samples in the columns as well as standard errors and p-values. The results in the table can be further visualized using volcano plots and heat maps, where colors represent the estimated expression level. Packages like ggplot2 and R and Matplotlib in Python are often used to create the visuals. The table can also be annotated using a reference annotation file, usually in GTF or GFF format to provide more context about the genes, such as the chromosome name, strand, and start and positions, and aid result interpretation. 14, 12, 13, 26. Functional enrichment analysis. Functional enrichment analysis identifies biological processes, pathways, and functional impacts associated with differentially expressed genes obtained from the previous step. It uses tools like GoSeq27 and PathView. 28. This creates a table with information about what pathways and molecular processes are associated with the differentially expressed genes, what genes are down or upregulated, and what gene ontology terms are recurrent or overrepresented. 14, 12, 13, 26. RNA sequence analysis explores gene expression dynamics and regulatory mechanisms underlying biological processes and diseases. Interpretation of images and tables are carried out within the context of the hypotheses being investigated. See also, transcriptomic technologies. Analyzing protein sequences. Proteome sequence analysis studies the complete set of proteins expressed by an organism or a cell under specific conditions. It describes protein structure, function, post-translational modifications, and interactions within biological systems. It often starts with raw mass spectrometry (MS) data from proteomics experiments, typically in MZML, MZXML, or raw file formats. 14. Beyond preprocessing raw MS data to remove noise, normalize intensities, and detect peaks in converting proprietary file formats e.g., raw to open source formats MZML, MZXML for compatibility with downstream analysis tools, other analytical steps include peptide identification, peptide quantification, protein inference and quantification, generating quality control report, and normalization, imputation and significance testing. The choice and order of analytical steps depend on the MS method used, which can either be data-dependent acquisition (DDA) or independent acquisition (DIA). 14, 29. Genome browsers and sequence analysis. Genome browsers offer a non-code, user-friendly interface to visualize genomes and genomic segments, identify genomic features, and analyze the relationship between numerous genomic elements. The three primary genome browsers, Ensemble Genome Browser, UCSC Genome Browser, and the National Center for Biotechnology Information, NCBI, support different sequence analysis procedures, including genome assembly, genome annotation, and comparative genomics like exploring differential expression patterns and identifying conserved regions. All browsers support multiple data formats for upload and download and provide links to external tools and resources for sequence analyses, which contributes to their versatility. 30-31. Sequence alignment. Example: multiple sequence alignment. There are millions of protein and nucleotide sequences known. These sequences fall into many groups of related sequences known as protein families or gene families. Relationships between these sequences are usually discovered by aligning them together and assigning this alignment a score. There are two main types of sequence alignment. Pairwise sequence alignment only compares two sequences at a time, and multiple sequence alignment compares many sequences. Two important algorithms for aligning pairs of sequences are the Needleman-Wunsch algorithm and the Smith-Waterman algorithm. Popular tools for sequence alignment include pairwise alignment BLAST, dotplots, multiple alignment ClustalW, PROBCONS, Muscle, MAFFT, and tCoffee. A common use for pairwise sequence alignment is to take a sequence of interest and compare it to all known sequences in a database to identify homologous sequences. In general, the matches in the database are ordered to show the most closely related sequences first, followed by sequences with diminishing similarity. These matches are usually reported with a measure of statistical significance such as an expectation value. Profile comparison. In 1987, Michael Gribskov, Andrew McLaughlin, and David Eisenberg introduced the method of profile comparison for identifying distant similarities between proteins. 32. Rather than using a single sequence, profile methods use a multiple sequence alignment to encode a profile which contains information about the conservation level of each residue. These profiles can then be used to search collections of sequences to find sequences that are related. Profiles are also known as position-specific scoring matrices (PSSMs). In 1993, a probabilistic interpretation of profiles was introduced by Anders Krogh and colleagues using hidden Markov models. 33-34. These models have become known as profile HMMs. In recent years, methods have been developed that allow the comparison of profiles directly to each other. These are known as profile-profile comparison methods. 35. Sequence assembly. Sequence assembly refers to the reconstruction of a DNA sequence by aligning and merging small DNA fragments. It is an integral part of modern DNA sequencing. Since presently available DNA sequencing technologies are ill-suited for reading long sequences, large pieces of DNA, such as genomes, are often sequenced by 1. Cutting the DNA into small pieces, 2. Reading the small fragments, and 3. Reconstituting the original DNA by merging the information on various fragments. Recently, sequencing multiple species at one time is one of the top research objectives. Metagenomics is the study of microbial communities directly obtained from the environment. Different from cultured microorganisms from the lab, the wild sample usually contains dozens, sometimes even thousands of types of microorganisms from their original habitats. 36. Recovering the original genomes can prove to be very challenging. Gene prediction. Gene prediction or gene finding refers to the process of identifying the regions of genomic DNA that encode genes. This includes protein coding genes as well as RNA genes, but may also include the prediction of other functional elements such as regulatory regions. GRE is one of the first and most important steps in understanding the genome of a species once it has been sequenced. In general, the prediction of bacterial genes is significantly simpler and more accurate than the prediction of genes in eukaryotic species that usually have complex intron-exon patterns. Identifying genes in long sequences remains a problem, especially when the number of genes is unknown. Hidden Markov models can be part of the solution. 37. Machine learning has played a significant role in predicting the sequence of transcription factors. 38. Traditional sequencing analysis focused on the statistical parameters of the nucleotide sequence itself. The most common programs used are listed in Table 4.1y. Another method is to identify homologous sequences based on other known gene sequences. Tool see Table 4.3. Lee. 39. The two methods described here are focused on the sequence. However, the shape feature of these molecules such as DNA and protein have also been studied and proposed to have an equivalent, if not higher, influence on the behaviors of these molecules. 40. Protein structure prediction. Target protein structure 3DSM, shown in ribbons with Kalfa backbones, in gray, of 354 predicted models for it submitted in the CASP8 structure prediction experiment. The 3D structures of molecules are of major importance to their functions in nature. Since structural prediction of large molecules at an atomic level is a largely intractable problem, some biologists introduced ways to predict 3D structure at a primary sequence level. This includes the biochemical or statistical analysis of amino acid residues in local regions and structural the inference from homologs or other potentially related proteins with known 3D structures. There have been a large number of diverse approaches to solve the structure prediction problem. In order to determine which methods were most effective, a structure prediction competition was founded called CASP, Critical Assessment of Structure Prediction. 41. Computational approaches and techniques. Sequence analysis tasks are often non-trivial to resolve and require the use of relatively complex approaches, many of which are the backbone behind many existing sequence analysis tools. Of the many methods used in practice, the most popular include the following: dynamic programming, artificial neural network, hidden Markov model, support vector machine, clustering, Bayesian network. Ruderal species. A ruderal species is a plant species that is first to colonize disturbed lands. The disturbance may be natural, for example, wildfires or avalanches, or the consequences of human activities, such as construction of roads, of buildings, mining, etc. Or agriculture, abandoned fields, irrigation, etc. A single-species ruderal community of Ditrichia viscosa on gravel near Petah Tikva Segula railway station, Israel. Compare with mixed species image below. The term ruderal originates from the Latin word rudis, meaning rubble. Ruderal species typically dominate the disturbed area for a few years, gradually losing the competition to other native species. However, in extreme disturbance circumstances, such as when the natural topsoil is covered with a foreign substance, a single-species ruderal community may become permanently established. In addition, some ruderal invasive species may have such a competitive advantage over the native species that they, too, may permanently prevent a disturbed area from returning to its original state despite natural topsoil. Features. Features contributing to a species' success as ruderal are massive seed production, seedlings whose nutritional requirements are modest fast-growing roots, independence of mycorrhizae, polyploidy, quantification. Ecologists have proposed various scales for quantifying rurality, which can be defined as a ability to thrive where there is disturbance through partial or total destruction of plant biomass. Grime, Hodgson and Hunt, 1988. 1-2. The rurality scale of Grime presents values that are readily available, and it takes into account disturbance factors as well as other indicators such as the annual or perennial character of the plants. See also a mixed species ruderal community on the site of Israel's Road 40, north of the Yarkon River, where topsoil was left after the road building activity. Edge effect. Hemerobi. Pioneer species. Restoration. Or agriculture, abandoned fields, irrigation, etc. A single-species ruderal community of Ditrichia viscosa on gravel near Petah Tikva Segula railway station, Israel. Compare with mixed species image below. The term ruderal originates from the Latin word rudis, meaning rubble. Ruderal species typically dominate the disturbed area for a few years, gradually losing the competition to other native species. However, in extreme disturbance circumstances, such as when the natural topsoil is covered with a foreign substance, a single-species ruderal community may become permanently established. In addition, some ruderal invasive species may have such a competitive advantage over the native species that they, too, may permanently prevent a disturbed area from returning to its original state despite natural topsoil. Features. Features contributing to a species' success as ruderal are massive seed production, seedlings whose nutritional requirements are modest fast-growing roots, independence of mycorrhizae, polyploidy, quantification. Ecologists have proposed various scales for quantifying rurality, which can be defined as a ability to thrive where there is disturbance through partial or total destruction of plant biomass. Grime, Hodgson and Hunt, 1988. 1-2. The rurality scale of Grime presents values that are readily available, and it takes into account disturbance factors as well as other indicators such as the annual or perennial character of the plants. Abstract. This paper presents a synthesis by relating the history of the occurrence, development and consecration of ethology, as independent branch of biological sciences. Throughout history, aspects of animal behavior were studied by many naturalists and zoologists. Many thinkers of antiquity have studied animal behavior long before the word ethology was introduced. For instance, Aristotle, Plato, Chrysippus, Plutarch have left written evidence about their concerns in their contemporaries' attempts theorizing concerning animal behavioral motivation. Ethology has its scientific roots in the work of Charles Darwin and of American, German and French naturalists, including Charles O. Whitman, Oscar Heinroth, Wallace Craig, H. S. Jennings, Georges Louis Leclerc, Comte de Buffon, Jean-Baptiste Lamarck, Georges Cuvier, René Antoine, Perchaud de Réaumur, Pierre Gassendi, René Descartes, Charles-Georges Leroy, Georges-Frédéric Cuvier, Pierre Florenz. Although many naturalists have studied aspects of animal behavior through the centuries, the modern discipline of ethology is usually considered to have begun as a discrete discipline with the work of Dutch biologist Nicolaas Tinbergen and by Austrian biologists Konrad Lorenz and Karl von Frisch, joint awardees of the 1973 Nobel Prize in Physiology or Medicine. In Romania, research regarding animal behavior have started in the second half of the 20th century. The works published by M. Beniac 1970, 1971, Elena Chenzbrand 1973, 1978, and M. Kochu 1980, 1982, and, mainly, works, Salandiu et al., 1998, Palika, Komand 1998, Mihai Kochu 1999, Akadenkai 2009 contributed to development and popularization of ethology in our country. Ethology is a combination of laboratory and field science. Ethologists are typically interested in a behavioral process rather than in a particular animal group, and often study one type of behavior, such as aggression, in a number of unrelated animals. Understanding ethology or animal behavior can be important in animal training. Considering the natural behaviors of different species or breeds enables the trainer to select the individuals best suited to perform the required task. It also enables the trainer to encourage the performance of naturally occurring behaviors and also the discontinuance of undesirable behaviors. Keywords: history, development, ethology. Introduction. Over the last few years, the desire to understand the animal world has increased dramatically. Animal behavior is now a rapidly growing field and many prior understandings of the behavior, characteristics and communication of animals are being rewritten. The big question, how and why do animals behave the way they do, is far from being comprehensively answered, but the emergence of a new generation of animal behaviorists is helping to begin the answering of this and other questions. The study and interpretation of the natural behavior of animals is a fascinating subject, and those who choose to study animal behavior are concerned with understanding the causes, functions and evolution of behavior. There is an increasing need for humans to take an element of responsibility towards animals and their welfare. Not just for domestic pets but for wild animals, who are having to adapt to new environments as a result of climate change and the effects that this is having on the natural habitats of some wild animals. On a more domestic level, there is a greater awareness among the general public of issues involved with the welfare of companion, performance and farm animals. Animals that are bred for food, animals that we keep as pets and those that are kept in zoos and aquariums, horses that are used in horse racing and animals that are used in films are all looked after by people with a good scientific knowledge of animal behavior. 45. Research Journal of Agricultural Science, 47 2015. Material and method. I made a short references incursion on the history, occurrence, development and consecration of ethology, using existing materials from foreign and our country literatures. Results and discussion. Ethology is the science that study animal behavior, usually with a focus on behavior under natural conditions. The term ethology derives from the Greek language ethos meaning character, and logia meaning the study of. The term was first popularized by William Morton Wheeler, an American myrmecologist, the study of ants, who in 1902 became the leading authority on the behavior of social insects, achieving particular renown for his studies of social behavior of ants. He was instrumental in the development of ethology and first popularized the term in a 1902 paper in Science. S. Lee, Charlotte, 2007. An earlier, slightly different sense of the term was proposed by John Stuart Mill in his 1843 System of Logic. Bourg, Julian, 2007. He recommended the development of a new science, ethology, the purpose of which would be explanation of individual and national differences in character, on the basis of associationistic psychology. This use of the word was never adopted. The overall study of the behavior of animals in their natural environment has a long history. An enormous amount of descriptive material, as well as some experimental material, was collected in the works of 18th and 19th century natural scientists, for example, in the descriptions of the German scholar H. Réaumur and the French scientists G.L. de Buffon and J. H. Fabre, and in the experimental work of the French zoologist G. Cuvier. It was on the basis of such material that the category of instinctive behavior was singled out and clearly defined. Ethology was directly influenced by the works of Charles Darwin. The great number of facts he collected about the behavior of animals in their natural environment made it possible to distinguish the basic categories of behavior, namely, instinct, learning ability, and the elementary ability to reason. Darwin also showed that animals' behavioral traits, like physical traits, are inherited and subject to change. Using instincts as an example, Darwin indicated the ways in which behavioral traits can evolve in the process of natural selection. The development of ethological notions was directly influenced by the research studies of various scientists, the Englishman D. Spalding, the American C. O. Whitman, and the German O. Heinroth, who demonstrated experimentally that certain forms of behavior are of innate origin, are uniformly manifested, and are peculiar to certain species. The modern discipline of ethology is generally considered to have begun during the 1930s with the work of Dutch biologist Nicolaas Tinbergen and by Austrian biologists Konrad Lorenz and Karl von Frisch, joint winners of the 197 The Nobel Prize in Physiology or Medicine 1973, for their discoveries concerning organization and elicitation of individual and social behavior patterns. The desire to understand animals has made ethology a rapidly growing field. Since the turn of the 21st century, many aspects of animal communication, animal emotions, animal culture, learning, and even sexual conduct that experts long thought they understood, have been re-examined and new conclusions reached. New fields have developed, such as neuroethology. Scientists and amateurs have studied animal behavior long before the word. 46. Research Journal of Agricultural Science, 47-2015. Ethology was introduced. For instance, Aristotle had many interesting observations concerning animal behavior. The study of animal behavior was taken up more systematically mainly by German and British zoologists around the turn of the 19th century. The great British naturalist Charles Darwin (1809–1882), in his classic book on the theory of evolution by natural selection, Darwin, 1859, devoted a whole chapter to what he called instinct. As early as 1873, the British amateur investigator Douglas Spalding recorded some very interesting observations on the attachment behavior of young domestic chicks to abnormal objects, a phenomenon that was later called imprinting, after the German Prägung. Karl Ritter von Frisch, the 20th of November 1886 to the 12th of June 1982, was an Austrian ethologist. His work centered on investigations of the sensory perceptions of the honeybee, and he was one of the first to translate the meaning of the waggle dance. His theory, described in his 1927 book Aus dem Leben der Bienen, translated into English as the dancing bees, was disputed by other scientists and greeted with skepticism at the time. Only much later was it definitively proved to be an accurate theoretical analysis. Riley, J. et al., 2005. Nicolaas, Nico, Tinbergen, the 15th of April 1907 to the 21st of December 1988, was a Dutch biologist and ornithologist. In 1951, he published The Study of Instinct, an influential book on animal behavior. In the 1960s, he collaborated with filmmaker Hugh Focus on a series of wildlife films, including The Riddle of the Rook, 1972, and Signals for Survival, 1969, which won the Italia Prize in that year and the American Blue Ribbon in 1971. Konrad Zacharias Lorenz, the 7th of November 1903 to the 27th of February 1989, was an Austrian zoologist, ethologist, and ornithologist. Konrad Lorenz is considered to be one of the principal founders of ethology, a branch of science that attempts to gain a deeper insight of behavioral patterns in animals. With Oskar Heinroth, the author of Die Vogel Mitteleuropas, he is held to have discovered imprinting, an especially rapid and relatively irreversible learning process that occurs early in the individual's life where auditory and visual stimuli from an animal's parents are needed to induce the young to follow their parents. A central concept complementary to imprinting is the innate release mechanism, whereby organisms are genetically predisposed to be especially responsive to certain stimuli such that imprinting will become fixed on the parents. age-of-thesage.org/scientist/Konrad_Lorenz.html closing parenthesis. Pierre Gassendi 1592–1655, in his Syntagma Philosophicum he undertook a comparative study of animal and human cognitive abilities and discovered they were logically similar. Both human and animal souls operated on sensory images to yield reasoned action. René Descartes 1596–1650 discussed the theory of animal behavior in his letter 1646 to the Marquess of Newcastle. French sensationalists continued to be chary of the use of instinct in the account of animal behavior. The attitude of Jean-Antoine Boyer, 17,131,764, a historian of animal psychology writing in mid-century, is representative. Georges-Louis Leclerc, Comte de Buffon 1707–1788, was a French naturalist, mathematician, cosmologist, and encyclopedic author. His works influenced the next two generations of naturalists, including Jean-Baptiste Lamarck and Georges Cuvier. Buffon published 36 quarto volumes of his Histoire naturelle during his lifetime, with additional volumes based on his notes and further research being published in the 2 decades following his death. Island ecology. Island ecology is the study of island organisms and their interactions with each other and the environment. Islands account for nearly 1/6 of Earth's total land area, yet the ecology of island ecosystems is vastly different from that of mainland communities. Their isolation and high availability of empty niches lead to increased speciation. As a result, island ecosystems comprise 30% of the world's biodiversity hotspots, 50% of marine tropical diversity, and some of the most unusual and rare species. Too many species still remain unknown. The diversity of species on islands is highly impacted by human activities such as deforestation and introduction of the exotic species. In response, ecologists and managers are directing attention towards conservation and restoration of island species. Because they are simple systems, islands provide an opportunity to study processes of extinction that can be extrapolated to larger ecosystems. Ecological processes on islands. Islands are attractive sites for ecological research because they provide clear examples of evolution in action. They show interesting patterns of colonization, adaptation, and speciation. Colonization and establishment. Islands are surrounded by water, and may or may not exist as part of a continental landmass. Oceanic islands arise due to volcanic activity or reef growth, and usually subside over time due to erosion and changing sea levels. When islands emerge, they undergo the process of ecological succession as species colonize the island. See theory of island biogeography. New species cannot immigrate via land, and instead must arrive via air, water, or wind. As a result, organisms with high dispersal capabilities, such as plants and birds, are much more common on islands than are poorly dispersing taxa-like mammals. However, some mammals are present on islands, presumably from swimming or riding on natural rafts that are washed away from the mainland. Of the species that arrive, only some will be able to survive in established populations. As a result, islands have fewer species than mainland habitats. Island populations are small and exhibit low genetic variability, see founder effect, but are isolated from the predators and competitors that they initially evolved with. This can lead to a process called ecological release, where a species is released from its ancestral community interactions and then colonizes new niches. Adaptation. In response to these changing ecological pressures, island species can become much more docile than their mainland counterparts, and may grow larger, see island gigantism, or smaller, see island dwarfism. Some of these unique adaptations are reflected in charismatic island species such as the Malagasy hippopotamus, Komodo dragon, or pygmy mammoths. Although, the giant tortoises of the Galapagos Islands and the Seychelles, the Galapagos tortoise and Albabrachles respectively, are sometimes given as examples of insular gigantism, they are now thought to represent the last remaining populations of historically widespread giant tortoises, i.e. gigantism is an ancestral trait that occurred in the absence of insular selection pressures. The collection of differences in morphology, ecology, physiology and behavior of insular species compared to their continental counterparts is termed island syndrome. 4.5. The Komodo dragon is an example of island gigantism. After immigration, birds, and some reptiles or mammals, tend to become larger and predatory. 6. Showing relaxed interspecific competition. 7. For mammals, small species will increase in size and large species will decrease in size. 8. This is referred to as the island rule, and is suggested to minimize energy expenditure. 9. Other adaptations to life on islands include increased poikilothermy, 9. Relaxed anti-predator behavior, 10. 11. And reduced sexual selection, 12. In animals, and loss of herbivore defenses, 13. And reduced dispersal, 14. In plants. Speciation. The formation of new islands and their isolation from the mainland provides many unoccupied niches for species to adapt to. Since immigration of predators and competitors is limited, many organisms are able to persist in these new niches. This results in a high occurrence of endemism, where species are unique to a localized area. For example, 50% of endemic bird areas are found on islands. 2. Endemism is often the result of adaptive radiation. 1. Adaptive radiation is when a single species colonizes an area and rapidly diversifies to fill all of the available niches. A common example is the assemblage of finch species documented by Charles Darwin in the Galapagos Islands. Darwin's finches exhibited adaptive radiation by evolving different beak sizes to exploit the diversity of seeds present on the different islands. The Palila, one of several endangered honeycreepers that evolved through adaptive radiation and are endemic to the Hawaiian Islands. Because the distributions of these populations are limited by their island habitats, they tend to have fewer individuals than their mainland counterparts and lower genetic variation. This, along with the behavioral and ecological factors mentioned above, makes island species more vulnerable to extinction. 1. Survival. The continued survival of species on islands depends on factors such as natural selection, genetic variation, natural disturbances, hurricanes, volcanic eruptions, and human-caused disturbances, introduced species, habitat loss. Human-caused disturbances tend to be the greatest cause of mortality, and understanding the causes of extinction facilitates conservation efforts. Human impacts on island ecosystems. The movement of humans to islands has led to rapid extinction of native island species either from hunting, habitat destruction, or introduced species. Hunting. Many large animals on islands have been hunted to extinction by humans. A well-known example is the dodo, once found on the island of Mauritius. 15. It evolved to become large, flightless and docile, and was subsequently driven to extinction by humans and introduced predators. Habitat destruction. The depletion of natural resources can have dramatic effects on island ecology. On Easter Island, the depletion of the forest by humans not only resulted in widespread loss of species, but also the collapse of the island civilization. 16. Easter Island has been a site of dramatic ecological change. Today there are over 500 million people on islands, all dependent on local resources either directly, traditional use, or indirectly, ecotourism revenue. Population growth and development result in heavy deforestation, pollution, and overexploitation. Overharvesting of ocean fauna is particularly troubling as yields of coral reef fish species are an important food source for island populations. Introduced species. Humans have contributed to globalization and decreased effective isolation of island communities, allowing for invasion of exotic species. This can have a profound effect on the native species. In Guam, the introduced brown tree snake ate nearly all of the native vertebrate species to extinction. Feral cats and dogs have also greatly diminished native vertebrate populations on islands, through both predation and disease. Introduced ungulates are another major threat, as they graze on native vegetation and can destroy entire forests. 17. Exotic grasses can outcompete native understory species and increase the risk of fire. 18. Lastly, social insects such as ants also cause major problems. 19. Non-native species introduced onto islands can have profound effects on an island's ecosystem, more so than non-native species introduced to continental land. Platenberg. The higher impact of introduced and non-native species on islands is largely a result of lower biodiversity levels. Platenberg et al. Biodiversity on islands is especially threatened by logging, hunting, fishing and plant gathering. However, islands' vulnerability to introduced species is also due to agricultural, economic, and health differences as compared to continental land. Russell et al. The smaller land area and population sizes of islands compared to continents create greater vulnerability to the impacts of introduced species. Russell et al. Introduced species negatively impact ecosystems through altered predator-prey interactions that can cause harm or even local extinction to native species populations. Towns et al. There are many examples of animal species such as birds, reptiles, and aquatic insects being harmed by the introduction of predators such as rats, cats, and ants. For example, seabirds on islands in Hawaii are impacted by non-native predators like barn owls and ants. At a colony in Hawaii, at least 20% of wedge-tailed shearwater eggs were taken by mynas, dark-plumaged birds of the starling family, Towns et al. Also in Hawaii, more than 44 introduced ant species, especially tropical fire ants, caused detriments to the growth of shearwater chicks, such as loss of tissue in their feet, Towns et al. Other examples of high-impact species to island ecosystems are the Cuban tree frog and cane toad, which were introduced to the U.S. Virgin Islands under various circumstances, Platenberg. The Cuban tree frog has been present in the USVI since arriving unintentionally on a cargo ship in the 1970s, Platenberg. The Cuban tree frog is known for its ability to survive under harsh conditions, and it is highly adaptable, as it will eat a wide variety of organisms, Platenberg. Native frog and anole lizard populations have declined, likely due to Cuban tree frog interference, Platenberg. In contrast to the accidental introduction of the Cuban tree frog, the cane toad was deliberately introduced to control agricultural pests, Platenberg. Similar to the Cuban tree frog, the cane toad is also omnivorous. Cane toad tadpoles compete for limited freshwater resources with the white-lipped frog, a native species, thus limiting their resource. Cane toad tadpoles compete for limited freshwater resources with the white-lipped frog, a native species, thus limiting their resources, Platenberg. In general, when the introduction of a non-native species results in extinction, the ecosystem experiences losses in some trophic levels, Platenberg. For example, vertebrate herbivores that are prone to extinction change the ecosystem function in plant communities. This phenomenon is seen in New Zealand, where the loss of bird species may have changed dynamics in avian-induced vegetation communities and impacted abundances of forest plants, Platenberg. Also in New Zealand, small mammalian predators such as rodents have little direct effect on vegetation but have greater effect on island faunas through extinction and displacement, Wood et al. Pacific rats, for example, were thought to be causing the local extinction of large, nocturnal ground-dwelling lizards, and they were assumed to have minimal effect on diurnal species, those that sleep at night, such as shore skinks, Wood et al. In places where Pacific rats were removed, however, shore skink populations rapidly changed, indicating that the rat's impact has been underestimated, Wood et al. The loss of a keystone species, a species that all other species in the ecosystem depend on such as seabirds, can also have significant effects, Towns. Seabirds are essential to the structure of the ecosystem because they transport large amounts of nutrients into ecosystems and burrow into soils, helping vegetation, Towns. However, since human settlement, seabird communities have been severely impacted on islands across the globe, Towns. While some exotic species may perform ecological roles similar to those of extinct species, there are many roles that cannot be fulfilled by other species. Therefore, similar exotic species do not offer complete replacement for extinct species in the community, Towns. Even if the number of species introduced to an island is roughly the same as the number of extinct ones, the ecological traits are not comparable enough to make up for losses, Towns. Global warming. Global warming is emerging as a strong cause of species loss on islands. This can be due to sea level rise, the intrusion of salt water into freshwater habitats, or species' inability to adapt to increasing temperatures and extreme weather events. Plant species are particularly susceptible. 20. In more isolated areas, such as the Southern Ocean Islands, indirect effects such as invasive species and global warming can play a greater role in influencing populations than overexploitation, pollution and habitat loss. 21. Sea level rise is a pressing concern for many island people. Trophic cascades. Human activities and the introduction of non-native species often cause trophic cascades, where direct effects on one species result in indirect effects on other species in the food web. An example is on Santa Cruz Island of the California Channel Islands, where DDT poisoning reduced bald eagle populations. This, along with an abundance of introduced feral pigs for prey, allowed golden eagles to colonize the island and replace bald eagles. However, the golden eagles also ate native island foxes. Fox population levels decreased to near extinction, while skunk populations increased due to relaxed competition with foxes. Island conservation. Conservation on islands. Since island ecosystems are self-contained, it should be possible to mitigate many of the threats to species. Ecologists and managers are working together to prioritize areas for conservation and to quickly design and implement action plans. Not everything can be put into a reserve, so it is important to first compile pertinent information and prioritize areas of concern. 22. Once an area has been chosen, managers must then acquire ownership and gain support. Local experts and indigenous populations should also be involved in this process. 22. Having clearly defined goals will facilitate the many necessary interactions between people and agencies. 22. Once a reserve is in place, managers can then practice adaptive management and do continued community education. On land, island conservation focuses on the protection of species and their habitat. In some cases, conservation can be integrated with agricultural production. For example, the acacia koa plantations and wooded pastures in Hawaii are anthropogenically altered ecosystems, yet allow connectivity between forest fragments and thus maintain higher diversity than would open pasture. 23. Other directions include habitat restoration, and eradication of introduced predators, ungulates, and exotic plants via hunting, removal or biological control. In marine ecosystems, there has been an increasing establishment of no-take reserves. This allows for the re-establishment of native species, and also the augmentation of commercially harvested species. 24. However, in both terrestrial and marine systems, these actions are expensive and do not always result in the desired outcomes. For example, some non-natives become keystone species and their removal can cause more harm than good to the ecosystem. To be more effective, managers of island ecosystems should share information and learn from each other's mistakes. 25. Lobster benefit greatly from the establishment of no-take zones on islands such as Great Britain, New Zealand, and Tonga. Island restoration. Island conservation tends to focus on preservation of individual species and their habitats. However, many ecologists caution that ecological and evolutionary processes should be conserved as well. 25. The conservation of island communities as a whole is closely linked to restoration. Active restoration on islands can be done for both animal species translocations, induced breeding, and plant species reforestation. Creating goals for restoration can be challenging because it is often impossible to restore the ecosystem to its historic or normal state, if that state can even be clearly defined. Restoration is never complete, as ecological communities